If you're evaluating software to support your next-generation sequencing workflows, this is worth your time. Choosing the wrong system isn't just inconvenient. It can derail your research, introduce risk, and slow down your lab. In this video, we'll break down what to look for in NGS software and why choosing an NGS LIMS that's designed specifically for genomics makes a measurable difference. We'll also look at where traditional tools fall short and what a more modern approach can offer. NGS workflows are among the most intricate in modern laboratories. These include workflows like whole exome sequencing for rare disease research, RNA-seq for expression profiling, and targeted gene panels used in oncology and pharmacogenomics. Each has its own complexity, data requirements, and integration points, from sample prep to downstream bioinformatics. Each sequencing run spans multiple steps, from sample prep and pooling to instrument setup and bioinformatics. Every action generates detailed metadata, produces large volumes of raw and processed data, and introduces decision points that must be tracked. You aren't just moving samples through a pipeline. You're managing barcoded libraries, plate layouts, reagent consumption, quality checkpoints, and terabytes of instrument output. Most of that needs to be traceable and auditable. Basic sample trackers, spreadsheets, or LIMS platforms built for simpler workflows often can't handle this kind of complexity. They may be able to record sample data, but they don't model the relationships between samples, libraries, protocols, and analysis pipelines. As sequencing volumes grow or new workflows are introduced, these tools become harder to manage and slower to adapt. Often, labs adopt systems that seem flexible enough on the surface. It might be a LIMS with bolt-on modules or a general-purpose tool that's been retrofitted to support sequencing. But when sample volume increases or workflows evolve, those systems struggle. Teams find themselves relying on manual handoffs or custom code to bridge the gaps. Scientists spend time chasing down results across disconnected tools. Analysts are forced to reprocess data that should already be accessible. And over time, small inefficiencies turn into serious bottlenecks. When your LIMS can't adapt to new protocols or scale with your lab, the cost isn't just operational. It's scientific. A system that supports NGS effectively doesn't just log samples. It understands sequencing as a structured process and can manage the relationships between samples, libraries, pools, sequencing runs, and results. Whether you're building somatic variant detection workflows or quality-controlled pipelines for library preparation and pooling, you need to ensure that every step, from QC checkpoints like RNA integrity to automated sequencing run setup, is fully traceable and adaptable. It should handle quality checks, track instrument metadata, integrate with informatics pipelines, and record every step of the workflow in a way that's traceable and searchable. Scientists should be able to configure experiments based on templates or protocols. Analysts should be able to launch downstream workflows without leaving the platform. Managers should be able to audit activity without needing to reconstruct events from logs or exports. In other words, the system needs to reflect the reality of how sequencing is done. There's been a lot of talk about AI in lab environments, especially for data interpretation. But in most systems, AI is an afterthought. It's a separate tool, disconnected from the lab's actual workflows. The approach is different when AI is built into the platform from the start. In this case, the assistant doesn't just analyze data. It understands how your system works and can take actions on your behalf. In Sapio's system, for example, Elaine is a science-aware assistant that can build experiments, configure workflows, launch analyses, and retrieve specific results, all through natural language interaction. You can ask it to find all samples that failed QC last month, create a new sequencing run from a stored protocol, or generate a new dilution series with updated inputs. Because Elaine is part of the system, not bolted on, it has access to your data, your logic, and your configurations. This means it can do more than answer questions. It can act as a collaborator in the lab, helping new users ramp up faster and letting experienced users move more efficiently. If you're looking for an NGS LIMS to support your workflows, the reality is this: not all lab software is equipped to handle this kind of complexity. The wrong choice can stall your research, burden your team with manual work, and create long-term inefficiencies. A platform that models scientific processes, integrates AI as a collaborator, not just a search bar, and supports flexible, scalable workflows can fundamentally change the way your lab operates. Before you commit to any system, make sure it reflects the way science actually happens in your lab. If you want to see what a modern NGS LIMS looks like in practice, explore a demo or learn more about how science-aware informatics can support your team.